Okay, welcome to Conversations with Faculty Part 2. Today we have Dr. Liliana Salvador, and then we'll have Dr. Alonso Favela, and then Juliana Gil Loaiza, and Dr. Alexander Bucksch. I'm probably butchering his last name, but yeah, we'll start with Dr. Salvador. Thank you so much for being here.
Thank you so much for having me. So I will start by sharing my screen and show some of the work that we do here in the lab. Let me just slideshow and share my screen. Can you see my slides? Yes. Okay.
Yes. Yeah.
Okay, great. So I'm just going to start. So the work that I'm going to talk about today, it is how can we go from computational biology to disease control, and how that actually focusing on computational measures can advance control measures in infectious diseases. So I am originally a computer scientist that has shifted to disease ecology, to animal movement during my PhD. And during my PhD, one of the I was exposed to the wildebeest migration, and also that cattle that occupies adjacent places to the wildebeest migration sites, they actually get malignant catarrhal fever. And so what the pastures do is that they move the cattle. So they try to predict the wildebeest migration, and they avoid those areas and move the cattle to other areas to avoid the cattle to get infected from the wildebeest. So based on this, and because I was studying animal movement at the time, I was really interested in how animal movement patterns could influence transmission in a multi-host system. So I worked at the University of Glasgow. I started working with that in mind. I'm sorry, I'm just trying to move this. Okay. With that in mind, I start focusing on bovine tuberculosis dynamics, and I have three main questions that we try to answer in the lab. The first one, it is, what is the role of cattle movements for infectious diseases transmission? And for that, we use some network simulation models to try to understand what is the impact of this. And so just for you to have an idea of why do we actually study cattle movement networks, because don't cattle live in the farm and stay there? So just for you to have an idea of the dimension of how they can move, I'm going to show you a video. And this video is based on Republic of Ireland in 2016. And you will see evidence. So a green dot represents a farm. And when we have an arch, represents a movement from farm to farm, or farm to slaughterhouse. So you can see that each one of these blue lines, it is a movement from an animal from the farm to the slaughterhouse, and the red circle and the red arches are from farm to farm. And these are exactly 6.7 million individual cattle movements that happen. And this is just in Republic of Ireland alone. I will just play this for a little bit longer for you to have a dimension, just an idea of the dimension. And here you can also see how many movements happen actually per day. And this is just an explosion. So I'm just going to stop here, but this is just an explosion of cattle movements that happen every single day from farm to farm, and from farm to market, from market back to the farm, and also from farm to slaughterhouse. So you can imagine that from a disease perspective, if one of these animals is infected, and then if it is not diagnosed properly, can actually spread the disease to other farms and across the entire country. So for that, we base this network. So each individual has a tag, and with that, we keep a record of all the movements and all the life history of these animals. And so, this is the system is called Cattle Tracing System Database. And with that information, we can then create these beautiful networks, and which are an ideal system to study infectious disease dynamics. And we developed large-scale individual-based models, and to try to understand and predict what are the areas of future risk for bovine tuberculosis transmission. And we consider livestock movements, we consider local base spread, which is based if the farms have been infected already, and also we consider a background spread, which is it is mostly by the impact of wildlife. And then we can perform our model predictions and try to understand where we have high risk areas for bovine TB, but also what are these transitional risk areas, the areas that are low risk and then become high risk. So the other question that we are interested in is what actually is the role of wildlife for bovine tuberculosis dynamics? And for that, we use next generation sequencing. So we have shown before, using whole genome sequence data, that is isolated from the bacteria that is extracted from these animals that get infected with bovine TB. We can actually study cross-species transmission patterns and infer how much and how often these animals have been transmitted to each other. A previous study that of our team has shown that elk actually has not have any impact of transmitting the disease to cattle and white-tailed deer, and it's actually in this area of study, which is Michigan, where the bovine TB, it is endemic in the white-tailed deer population. We have shown that it's actually the white-tailed deer that is maintaining the disease, and it is responsible for transmitting to cattle and also for elk. So cattle and elk in this situation, they are mostly dead-end hosts. So, but there have been other animals in the area that have been infected with bovine TB. And so what is the question is, are any of these animals have the potential to become a new reservoir of the disease, which means if they can harbor and maintain the disease within their population and transmit it to other species. So we're using the same techniques, whole genome sequencing. We are able, from the bacteria that is extracted from these different species, we are able to estimate who infects whom at the farm level, and also who infects whom in the wild. And this is just an ongoing project, but it will reveal or give us information about what is the actual role of each one of these species in the transmission of this disease. We also are very interested in understanding host pathogen and host adaptation and pathogen-host interactions. And for that, we have a project on transcriptomics to identify the signs of bacteria adaptation to each one of these hosts. This is a collaboration that we have with USDA, and we hope that we will have a list of differential expressed genes that will be able to determine differences across species. And for the last question that our lab focuses on, it is how to control zoonotic TB. And for that, we use One Health approach. So bovine TB or animal TB can infect any mammal, and can, as we have shown here, it can cross the species barrier, but can also infect humans. And the way that infects humans can be either through indirect transmission by ingesting contaminated milk or other dairy products that have been infected, or actually direct through airborne transmission from infected animals, as well as between people, or also just by, for example, dressing a deer that is contaminated, and then by mistake we will cut our skin. So that will be a way of getting infected as well. So unfortunately, zoonotic TB, it is prevalent in areas where bovine tuberculosis is prevalent in livestock, and affects mostly neglected and living in low-income, rural, high TB burden areas. So and a lack of milk pasteurization, as we can see, for example, in this figure, it is one possible transmission route. And in many countries, this is still a very common practice. So we work very hard in trying to break the chain of transmission between animals and within animals, but also between animals, livestock and wildlife, and of course, between wildlife, livestock, and humans. So we run the computational zoonosis laboratory in ACBS, and you already understood that this is a very interdisciplinary research at the interface of microbiology, infectious diseases, and bioinformatics. So we perform molecular pathogen surveillance, bacterial zoonosis, bacterial phytodynamics, host adaptation, and food security. And these are some of the techniques that we use to be able to answer the questions that we are interested in. And these are some pictures of some of the lab members. These are lab members and also some of our collaborators. And currently here in Arizona, these are our hard workers in the projects. And I will be very happy to answer any questions, and feel free to email me also at any time at lilianasalvador@arizona.edu. Thank you.
So we have a few minutes for questions before the next presenter starts. Oh, looks like we have a question. Andrea? Feel free to oh, you were clapping. I'm so sorry. That wasn't a hand up.
It was not a question. It was like clapping.
Thank you.
Yeah, I just realized. I'm so sorry.
That's okay.
Well, if we don't have any questions from the group, we can move on to the next presenter.
Great, thank you.
Yeah, thank you so much. Dr. Alonso Favela is up next.
Howdy, let me set this up. Excuse me one second. I feel like I forget how to use Zoom every time I use Zoom. Does this look really weird right now? Is there like huge brackets on the side?
Yeah, there are brackets on the side.
Okay, let me try to fix that. I apologize.
It's not bad to look at though. It was still okay.
It's because I have a giant, insane monitor. Just don't want it to like mess up your thing. Okay. That's okay. Second to find the Zoom again. Okay, does that now look normally scaled?
Yes.
Great,
Yeah,
awesome,
good.
thank you so much. Yeah, thank you for inviting me to talk. I'm really excited to talk to you guys about my work and my research and the projects going on in my lab. So I just started at the University of Illinois in January. I was primarily trained as a microbial ecologist. So I'm really interested in the diversity and complexity of microbial communities on this planet, and more specifically interested in the microbial communities that are related to agricultural systems and the agricultural microbiome. And my lab primarily focuses on 3 central themes, with the ultimate goal being the incorporation of microbial interactions into our human-made systems. So in most sort of cases, we don't think about how complex microbial communities shape our day-to-day lives, but in reality, they're playing really fundamental roles in different parts of society, from our food to our agroecosystems to our health. And in my lab, I really focus on how they shape plant interactions and things in the agroecosystem. And I have 3 central themes, the first of which going into the plant genetic mechanisms of microbiome interactions, the consequences of these microbial interactions, and then how humans have shaped this agricultural eco-evo dynamics in the agroecosystem. But what are microbiomes? So microbiomes are ecosystems of microorganisms that live together in a community. These communities consist of bacteria, archaea, protists, fungi, viruses. They're very diverse. They have organisms from different kingdoms across the phylogeny of life, and they all interact together in close proximity. Microbiomes exist across a bunch of systems, from marine to terrestrial systems. They exist in soils, water systems, humans. So we have our own microbiome on our skin and our face and our guts, and they exist in association with plants as well. So they're really omnipresent across these systems and are really understudied compared to their presence in these systems. And why do we care about microbiomes? So microbes drive almost every food chain on Earth. They play central roles in nitrogen fixation, decomposition, nitrification, and denitrification, just to name a few of their functions. So they do a lot of things in the environment, in soils. Related to us, they play roles in our health, directly shaping our weight, how clear our skin is today, our mental health, and our sort of disease health. There's a lot of really interesting work going on right now on the gut-brain axis that basically is showing that the microbes that are existing and living in your gut can shape how anxious you are, how fearful you are, all your different behaviors. And so we really depend on all these activities of these microbiomes, and we're just really beginning to understand their functions and how they exist in these systems. And I am particularly interested in how they alter plant productivity and their functions in soils. And that's sort of what this point is. My group is really interested in starting to develop that foundational knowledge that microbiomes play in plants, in their ecosystems, and how they potentially interact with human health and nutrition. And just to get into those very specific themes that I was talking about, that first theme of plant genetic mechanisms involved in microbial interaction, the sort of questions we're asking how does the plant actually shape the microbiome in the soil? So plants at their root surfaces, they're exuding and putting out a bunch of chemicals into the soil system that has different effects on different microbes. Some really like that signal, some really dislike that symbol, and that shapes the outcome of that composition. In this theme, we're trying to understand what genes are underlying that selection, and can we select those genes to have specific microbial outcomes that benefit the agricultural system. And some research that I've done in that in the past is I was interested in looking at sort of how have we already changed the microbial interaction that plants and soils have? And the way I tested that was by looking at maize. Maize has been under selection by humans since forever, really. But I studied modern maize and created a chronosequence of maize lines that started in the 40s all the way to the 80s. And I saw how these plants differed in their microbial interaction. And what I found across that sort of time sequence is that the plants were selected to have less mutualistic interactions with the microbes in the soil. And we think that this is the case because of the soil environment changing over this time period. This is the same time period where we started applying fertilizers to soils and really started replacing that microbial interaction that would have typically happened between the plant and microbe, and replacing it with our fertilizers. So maize has become less mutualistic with nitrogen fixers. And this is the NIFH, the nitrogen fixing gene. We sequenced it, and then we looked and compared how these populations differed in the roots. The second sort of theme in the plant is really getting at understanding the consequences of this microbial composition change. So if you can imagine a community of microbes that has, 1,000 different unique species in it, different shifts in that community are going to have different outcomes. So here we're really trying to understand what microbes changing in the microbiome are important to predicting the function, and how do they relate to the cycling of nitrogen, carbon, and water in the soil system. So this is the idea that different microbes have different functions, and how they interact with each other shapes the ultimate outcome of those functions. And some research that I've done in this is I studied teosinte and maize. Teosinte is the wild progenitor to modern maize. And I grew them both in the same field. And then I measured some of their microbial functions. So nitrification and denitrification are two different processes that alter the flow of nitrogen. Both of them result in the loss of nitrogen. And in that study, we saw that teosinte, the wild progenitor, actually had a heightened ability to suppress these microbial impacts in the soil that would be harmful to sustainability. So these activities actually result in the loss of nitrogen from the soil system, and teosinte the progenitor, the wild relative, actually stop those activities happening in the soil system. So here we're seeing that plants actually have the ability to regulate the functions of these microbial communities. And we're really trying to understand, okay, what microbes are important to the shift? How are plants doing this? And then how do we incorporate this back into agriculture so that we have less leaky agricultural systems that are productive and not destroying this planet? And then this final theme, the theme that's sort of I think the most exciting is this sort of idea that humans have been changing the soils and the evolution of microbial communities over long and large timescales. And here we try to study it. So here we're trying to treat microbial communities like normal ecological communities and try to understand how extinction, evolution, and invasion are playing out in these microbial communities. And we're actually using agriculture as a tool to understand these questions because we have such good records of agricultural effects on soils and on these systems. And these are the sort of questions related to it. How do our activities change it? Are we losing specific microbes through our activities? And can we then reincorporate these microbes back into these systems? And some of the research that I've done on this, it's actually have been on the Morrow plots. This is the oldest experiment in North America. It's an agriculture experiment. They basically have been growing the same plots over and over again for 100 years. They started in 1876 and have gone on to today. I was lucky enough to collect samples from these archives and sequence them and measure the microbes that have been in these soils from the beginning to the end. And this is more work that I would like to continue going on Arizona and collecting more archival samples from this region and trying to see how our agriculture has been changing these soil microbial communities. But one of the big findings from this study is that using sort of metagenomics and sequencing, I assembled genomes and I looked at what changes were happening in the genome of those microorganisms. And from that, we were able to find that the actual fertilizer treatments that are being applied in the field are selecting for different genetic regions in these microorganisms. So it seems like here manure was selecting for specific alleles, contigs, and genes in the genome of the single microorganism, while the lack of fertilizers were selecting different alleles. So it seems like these soil microorganisms, these invisible actors in these systems, are being selected on by our treatments, by our agriculture, and we aren't even really being aware of it. So this is really starting to open the door of how we should start thinking about these microbes in these systems and how we can sort of improve our interactions with them to result in more sustainable outcomes in the agricultural system. Anyways, so those are sort of big, broad brushstrokes. We could have gotten to really specifics, and I'm happy to address more questions if you guys have very specific questions. But I think if you come and work with me and participate in my lab, there's a couple of skills that you're definitely going to have the opportunity to build. You'll have wet lab training. You'll have some bioinformatics and coding training. If you're lucky enough, you'll be able to learn how to plant breed and take care of plants. I don't know. I think plant breeding is really fun. It's kind of a lost art form. Enzyme assays, microbial enzyme assays, chromatography, working with soils, and sort of experimental design. Yeah, so we really need to start understanding how humans are shaping microbial communities at a granular level. We've been shaping them for a really long time. Here are a couple examples through history of how we've been shaping soils and the microbes that exist in them. And we really have to get specific to improve the sustainability of soils. And you know, these soils are threatened. Right now, 52% of all of our food producing soils globally are classified as degraded, and 90% could become degraded by 2050. These soils are dropping in quality, and we really have to understand why and have to really look backwards to understand and get the lessons from this. Yeah, anyways, more bad stuff about soils. We're not good at agriculture, but this is, I really believe, by incorporating a microbial understanding, we'll be better equipped to address some of these challenges. And these are the sort of central themes in my lab. And I think, oh, there's a bunch of other stuff. So these are the central themes in my labs. We're interested in genetics, microbiomes, ecosystems, how these all interact to functional sustainability. And I should be good for questions. Thank you so much.
Thank you. That was awesome. Any questions from the group? Don't be shy.
No questions?
All right. Thank
Okay.
you so much, Dr.
Yeah.
Favela.
Thank you so
Okay.
much.
We're going to move on to Julie Gil Loaiza. Yeah. You're muted, Juliana. Of
Sorry, thank you.
course.
As always. Uh, um, Can you hear my screen now? Hello, can you hear my screen? Can you see my screen?
Yeah, we can see your screen. You sound a little bit quiet.
Okay, I'm gonna try and check the microphone volume.
Oh, now I can't hear you at all.
Hello, can you hear me?
Still very quiet.
Hello,
Sorry.
can you hear me now? No. Hello. 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 Can you hear me?
Still very quiet. I'm so sorry.
I just have to with the call. Okay, I'm gonna. Can you hear me now? If I talk.
Oh yeah, that's getting much
Okay.
better. Yeah.
Okay, I don't know what is going on, and the microphone controls don't go. Okay, so my name is Juliana Gil. I am a research scientist.
Okay, I'm so sorry. It's not better. I'm so
Okay.
sorry.
It is the case, the next essentially, it starts, well, I try to figure it out now. If it's okay, the next, presenter, we can start and try to
Oh,
figure it
sure.
out.
Yeah,
Sorry about
that's good. Okay. Yeah, so we'll move on to
Thank you,
Dr.
Doc.
Bucksch.
Okay, you can certainly hear me, right? Okay, wonderful. Share. Share everything and do this. We should be good now, huh? Okay. Awesome. Hello. Okay. It's a bit of a surprise to be now the presenter, but we are very much interested in my lab in how plants play together. Call it like that. They're just team players. And what is a little bit different, so I'm not a biologist at all. I studied once math and computer science, more or less. And what I do is called phenomics. It's a phenomics-driven approach. And I simply thought I first tell you what that means. So what is a plant phenomicist? So you can do that with every organism, but we are doing that on plants. And basically we are doing formal systems like, a sensor is a formal system. A computer program is a formal system, basically something that you could also express in math. And then we use these systems to observe and discover phenomena in nature and try to understand them with a link to plant life. Very easy as a definition. But yeah, impact-wise, we are really interested in what is happening on this planet, and that's what we observe. So if you look just very broadly here, there on the left, there's a forest, right? This is a big carbon sink. All this CO2 in the air goes in there. We have the soil that also Alonso just mentioned. It's another big carbon sink, and we have the ocean, right? So these are the three biggest ones. Now, what we do with these is this: we cut down forests, especially the Amazon forest, and we see, and you can even observe, there's not much green anymore, right? It's simply basically is degraded land. Similar, what we also just saw, yeah, the soil becomes really, really degraded and more like desert-like, and the ocean comes into the landmass and just fosters this salinity everywhere. So these are all degraded lands. There's not much nutrients in it. But what we want to have is, yeah, land that is a little bit green. So how to get there? So the first viewpoint that we take that is a bit different than typically discussed is we simply say, okay, resources are sparse and a little bit far away from the root and the plant. That is for us a degraded soil in general. All the details we don't care at this point so much about. And the other thing we think is, hey, if it is a good soil, there are lots of nutrients or lots of resources. Water is also a resource. and it's closer to the plant, and it's easier to access, and it's more abundant. Well, if you look into literature, you will see roots. When we look at plants below ground, there's always just one root described for every kind of resource deficiency that is out there. So drought, you see here as an example of, I think, corn, that is the example, becomes very deep and thin, and otherwise others become very shallow and wide. But it's basically just one phenotype that is out there, one structure you could observe. And what we did is then say, hey, why not take looking at all the roots in a field, in a real agricultural field? So how does this look like? So that is the first root of a full field. And if you look through that, there's definitely more than just one of these root structures, or we call it root architectures in the field. It's kind of strange. You learn from the book that there's only one, but if you look at them, there's so much out there. So that's the first observation that is very simple to do. And yeah, you should wonder if you see that. So what is underlying that is simply that all these field evaluation that we previously did, probably over the last 100 years or so in ag research, they really rely on sampling just what is most common. So that what is dominant in the field. We never really looked at complete populations in the field and what is happening there. So what we did is we took this field and took it out of each other. So we came up with a method. How can we compare these architectures and quantify them? And what came out of it is that for this example here, which is a common bean, like a pinto bean, for example, it's always 5. There are always 5 architectures. It doesn't matter if you put it into drought, into other nutrient deficiencies, which year you take these samples, always 5 architectures, different ones. The only thing you see is that how often an architecture is present, that changes. So if you look at WW means like well-watered, and WL means water limited, kind of a drought, you see that there are certain shifts in the fractions, how often a certain architecture is present there. So if you look at the most dominant one, what has been always sampled, we actually base our conclusions always on about 30% of the variation that is out there in a field. That's where we got our knowledge from, and that's where the books are written on. So that's kind of what you get taught in your classes right now. But let's go one step further. We know that there are fractions. There's something to observe, right? We know these fractions are changing, but what is the function? So we came up with another method to match functions directly to these architectures. And what came out, without going into the method right now, which is a big deal actually to develop for us, these methods, it's these architectures, they correspond to certain resource types, I call it now. So these arrows simply denote different kind of mobility in the soil of resources that they specialize on. And then you have resources that just are very deep or very shallow in the top. So like phosphorus, for example, or nitrates. Nitrogen is more like a mobile resource. Water is a mobile resource. And water can also be just accumulate on the bottom. But basically, that's where these roots really specialize. And then I didn't mention that before. So we basically have a different curve for each of these architectures to distinguish them and to compare them. So that makes it quantitative. So that's nice. So we can distinguish that. We have a function. Let's see if this function actually does something in the real world. You want to go, we really collected everything in the field. We want to go back there and see if there's an influence. What we did is we took 2 of these. These are actually pinto beans, both different varieties. One from Chile. The other one is from, I think, southern Mexico, the SEQ7. And the DOR is the blue one is very much drought adapted, and the other one is very good for topsoil resources like phosphorus. So non-limiting conditions, yeah, they do pretty good, have same amount of seeds. If you put it into drought, the SEQ that is not adapted to drought really drops down. Now, let's put them together and say, hey, can you help each other? So we put them in a chessboard next to each other and see what's happening. Happens is, yeah, the drought adapted one does well, no change. The other one suddenly starts going up, has almost double the amount of seeds. And under drought, exactly the same. So we can actually compensate for the yield loss and even get more yield than we thought than we had before in non-limiting conditions, just by mixing the right varieties together and let them interact with each other with their strength. So it kind of gives you a feeling that plants might be not are more driven towards helping each other as a population, and everybody has a certain role in there for the population benefit, which is a theory that is simply out there. Okay, we look deeper back to these curves and saw, okay, there's a reason for that. It's not just putting something into the field. There's actually really an architecture arising that hasn't been observed before. So the SEQ suddenly changes one of the architectures completely and it has a different function that is very much related to like topsoil, but a little bit under the topsoil uptake. So that's where the boost is coming from. I thought that is just one nice example quickly to show you, okay, that's one of the type of studies we do where we just start observing with a lot of methods. I wanted also to let you quickly see what is in the background, some of the methods that we really developed to start observing these kind of phenomena. So one of them that we have ongoing is now this. It's a PhD in my lab, it's Will. And he came up with a pretty clever method to look at what two root systems do together. If you look on the bottom here, these are two root systems, but if I look at one of these roots in this system, I cannot decide by eye, even not be able to trace it back where it belongs to. So he came up now with a method to label them in color. It's basically a dye that we pump into the roots here over these, in the second picture, you see these pipes going into the root systems. But yeah, they get different colors, and that enables us to use machine learning to take them out of each other and measure. So because you need to distinguish that, you could not label that by hand. That's a very physical technology we did. Another thing we did is we thought the first example was all on 2D. We developed a 3D scanner, and that was about, I think, 4 or 5 years ago that we came up with this idea. It's just cameras going around a root, and it's optical. It's much cheaper than any whatever X-ray or MRI that is in the $500,000 million range or more. So that is just $2,000. So we can scale that up to many roots if you have enough people putting roots in there. That happened, and it even got very light. So that's an undergrad in my lab. That's Ruben, and he is part of the team that built this 2.5, maybe 2 kilogram scanner that now does the root scanning for us. The question is, I talk here about root scanning. What comes out of this? What comes out is that. It's a 3D model of the root. It's basically looking like an X-ray, right? That costs you $500,000 or $1 million. But you can start analyzing every tiny little branch in this root system. Just for $2,000, right? Probably a bit cheaper in a few years. But yeah, we get a decent detail that we can analyze and make statistics on, start thinking about why is the phenotype what it is, why does the root simply look like it is, and what does this has to do with a function in a whole field or on the whole population. Okay, I think that's pretty much what I wanted to show you as examples. It's more going on in the lab. So sure, we are interested in the genetic basis of how they start cooperating with each other and helping each other. We're just starting doing large-scale experiments on that where you can be involved. So that is currently planned. Actually, next week it's with like 40,000 seeds planted at the campus farm. If you want to come along and see how that looks like, will be over the summer that we work on that. There will be more genetics coming into it. We're also involved in developing non-destructive sensors of these curves. So there's a problem in the soil. There's so much iron, you cannot just put any electromagnetic wave on there. So we have a way to do that non-destructively with fibers that is currently developed, still in a pot, but one day it will be out in the field. So that's a way to get involved. And then we have another way to get involved with our research on multi-scale models. So how does root-root interaction go from one root to the other, but also across different scales? So there's not only the transmission that they know who is on the other side, there's also how is the anatomy reacting to that or these kind of things. So lots to play with, all in a big context that they actually play together. And yeah, some funders, there are some around. So that was in my previous university. I'm just here since a year. It was actually funded heavily by an undergrad institute. So there are many of them involved in here. And that's just the current lab. Pretty many people. Yeah, many undergrads, Ruben, Veronica, Max, they're all there. Yeah, here the Julius is also there. They're all undergrads in the lab that make that happen. Many collaborators all around the world. So there's lots to do with language barriers if you're interested. And that's it. That's all I wanted to say. Thank you.
Thank you so much. Any questions from the students in the room?
You're at lunch.
Thank you so much, Dr. Bucksch.
Sure. Yep. Stop
Cool.
sharing.
I think we will then move back to Dr. Gil Loaiza.
Hello, can you hear me now better?
It sounds so
Awesome.
good.
I don't
Thank
know
you
what
so
was
much.
okay, I'm going to hang up the call. So we don't have any feedback or anything. Thank you very much. Thank you, Dr. Bucksch, for taking over my spot. Appreciate it. While I was dealing with my technical problem with, as always happens with Zoom. Okay, can you see my screen and hear me well now? Okay, perfect. So, my name is Juliana Gil. I'm a research scientist at Dr. Laura Meredith lab, and she cannot be with us today. She's on a sabbatical, but I'm here to tell you about the research in our lab, which is basically it focuses on microbial drivers in soil that contribute to biosphere-atmosphere gas exchanges. And in this figure, what I want to show is how we approach our work at the lab. So we aim to understand biological and ecological relationships in the soil, considering the complexity of the soil as a matrix. And then to achieve this, we develop or utilize tools such as sensors, gas probes, to measure gases at a high resolution, then trying to be less disrupted as we can of our system. And then we gather all this information, and through the application of data integration methods and different omics and soil sampling, we draw mechanistic conclusions and scale our findings to achieve a comprehensive understanding of our systems. And we basically want to know who does what and why. In our research project, we typically research projects typically consist of two components. One is the lab research, which is a more controlled environment, and a field research, which we work like in a real world setting to gather data that validate our lab findings. So to scale up our projects or scientific questions from the lab to the field, these enable us to understand trace gas interactions across different scales and complexities. And it also provides like a more practical application perspective. So basically, we want to bridge the gap between the theoretical research and what are the real world applications. The latest grant, led by Dr. Meredith and awarded by the Department of Energy, is a collaborative project with Lawrence Berkeley National Lab and Adam Bay Area and Aerodyne Research Company in Boston, Massachusetts. And what we want to do in this project is we want to study what the VOC impacts on soil carbon. So VOCs, or volatile organic compounds, are prevalent but are overlooked part of the soil and in microbial systems. About 20% of the metabolites or the compounds in the soil are in a gas state. And most of the studies, they just focusing on those that are non-volatile. And also non-volatile metabolites, so they overlook the VOCs and the missing a significant component of the soil. So the overreaching goal of this project is to characterize and quantify VOCs as direct or indirect contributor of the soil carbon stabilization. I see some. Okay. Okay, sorry, I was just checking the chat in case there was any questions. Let me see, it's not moving. Okay, so as I mentioned before, usually we have a lab and a field component for this project. And our field component, we aim to identify VOCs consuming microbes and understand how those VOCs, so the VOC can be stabilized in the soil. And during this process, we want to see changes in biotic and abiotic soil factors over time and what will happen when we add a specific VOCs into this system. So to achieve this, the lab team is gonna be creating an automated system soil incubation in what will be added VOC labels that are labeled, and that label will allow us to track what is happening with the soil. Hello? If there's any question? Okay. So the team are going to be developing the system then with different types of soil, agricultural soil, forest soil, and also we're going to use silica or sand as a control. And additionally to that, we're going to have a VOC analyzer who will allow to have real-time online VOC measurement concentrations. And then we're going to do some soil sampling and do some processing and using next generation sequencing, metabolomics, and then we're going to integrate all these data to answer all these questions. So to give you an example or kind of view how is an automated experimental setup in our lab, this is soil incubation experiments that we have at Aerodyne Research. We build all these incubation jars, and here this is the Focus and mass spectrometer that allow us to measure the concentrations of VOC in real time. Here in the back, you can see some components that allow to an automated system. And as I mentioned, we love to make our systems more complex or scale up. So in the back in here, you can see how we scale those small jars to 1 meter length column. We wanted to know, okay, what is happening with the VOCs on a soil profile. So then the full component of this experiment will be at Maricopa Agricultural Center, in where we want to know how the VOC profile in the rhizosphere changes with different sorghum genotypes. And also, for example, how if the genotypes have a larger root, this will be that there's a greater VOC carbon contribution of these specific genotype to the soil carbon stabilization, just as an example. So we really want to know what is happening with the VOCs and the carbon cycling transformations with the different sorghum genotypes. And for this, we're going to be using the gas probes that the lab developed, integrated with the VOC analyzer for online real-time VOC measurements. And we're going to take samples to do some soil processing and genomics. So you might say, okay, the way or the how is similar to the lab. So we went to Maricopa Agricultural Center in 2021 for a nitrogen cycling project, although the objective was very different. It was different. The steps are going to be basically similar or the same. So we go to Maricopa Agricultural Center using the specific area that they have with their scanalizer to take different data about the different sorghum genotypes. And we're going to setting up the probes, do some field preparation, do soil installation, and then once the probes are in the soil and the seeds during that growth period of the plant, the team is going to be learning or taking soil, plant, and root sampling, doing some sample allocation, which is understanding what sample, how much of that sample goes to what specific downstream analysis, and how is the cycle of managing that sample. There's going to be a lot of instrumentation, coding, troubleshooting, gas sampling, and there's going to be also the opportunity to visit Aerodyne in Boston and help at Aerodyne in one of our lab experimental setup, as well as getting to know how the field is kind of work and what will happen and how we can answer those questions and based on the different sorghum genotypes. So at this point, you might be thinking, okay, why do we put so much effort to try to understand VOCs in soil? Like, why this work is important? Well, we really think that VOC carbon play an important role in the diversity and the quality of the carbon stabilization in soil. So we try to answer this question, we will have a better understanding of that carbon cycling and the microbial system in the soil. Then the practicality of these, it will be that we will have a better understanding of the stabilization of carbon's input from the VOC into a more stable soil organic matter pools, which is important to predict carbon sinks under climate change. Therefore, it may help developing new mitigation strategies and reveal new opportunities to manage, in this case, agricultural soils to increase carbon sequestration. And in general, just having this understanding, it will help to create a new framework for carbon pools and being considered as in the carbon models. And with this, thank you for being here. I'm open to any questions you have. If you want or need, or if you're curious about any information or about all the different projects that we have going on at the lab, you can visit our website. You can follow Dr. Meredith on X or Twitter. And yeah, I'm open to any questions that any of you might have.
I have a question. How often are you going out to the Maricopa Ag Center?
At the beginning, the first is going to start in April, so very close to the planting date when we're preparing the soil. That will be very often. We usually, the team will go every day to prepare and make sure that everything is ready before all the irrigation starts, because all the probes need to be in the soil before the seed planting. So, and also we need to make sure that all the sampling system is working and it's not just to make sure that we have the baseline of the first days of irrigation before the germination of the seeds. But then it will be just basically troubleshooting, maybe changing some gas tanks, or so it will be depending on the needs. But then there will be always like a senior or a postdoc or a PhD student going with the other undergrads.
Very cool. Very cool. All right. Any last questions from the students in the room or from the group? No? Okay. Well, thank you so much to all of our presenters. We really appreciate you taking the time to present your work. And like I said, we will be posting this online, and we'll be sending out a reminder to our students who are still looking for mentors to watch those videos and to hear from you and to contact you. But thank you again for being here.
Thank you.
Thank
It was
you.
very interesting to listen to everyone's research. Thank you so much for sharing.
It's been so cool.
Actually, we might be reaching out to some of you. And to all those very interesting projects and developments that you're working on. So thank you.
Thank you. Have a good day.
Yeah.
Thank you. You
Bye-bye.
too. Have a good
Thank you.
good rest of your
Bye.
day. Bye.